And then another commonly used DNA marker technique is the STRs, or short tandem repeats. They are also called microsatellites or simple sequence repeats. Simple sequence repeats are in the DNA of any plant. We have gene sequences. The sequences within the DNA which are responsible for production of any kind of protein, which transcribe and translate into proteins, those regions are called exons. And between these exons, between these gene sequences in any organism's DNA, there are regions called introns, and these introns have certain repeats or variable repeats. These or tandem repeats, whatever they are called, these repeats will be like a dinucleotide, two nucleotides, for example C and A in the DNA molecule ATGC. These are the four nucleotides that keep repeating in different combinations and permutations. So supposing, for example, a dinucleotide CA, that keeps repeating many times, and the number of times it repeats also varies. So in these intron regions, these repeats, they are different. The number of nucleotides that keep repeating are also different. What nucleotides keep repeating are also different. And again, the number of times that they repeat are also different. So based on this difference, however, these repeats are present in all the introns. So based on this these differences, we design primers and then we amplify these repeats. So the primers are designed based on the flanking regions of the repeats, and they are bound to the DNA, and these repeats are amplified. Many copies are produced, and these repeats, after amplification, when you electrophorese them, each of those samples will have a different molecular weight, and then the banding pattern is different. And this SSR markers or microsatellites, and most commonly when it is used in human identity testing, this is the most commonly used method. It is called short tandem repeats. However, when it comes to trees, when it comes to timber samples or any species, if it is clonally similar, then it will not be accurate. All the DNA markers will not be accurate. Though other apart from these techniques, though other DNA markers are also available in the case of identification, especially timber identification, none of the other molecular markers serve the purpose, whereas these molecular markers are used. These profiling techniques are used, and the most commonly and always when it comes to timber identification, it is what species that we have to identify, and that is sometimes as sir has also described earlier, it will be like during illegal felling of the trees, for example during theft or timber trafficking, and also timber samples which are associated with other crimes also need to be identified, and sometimes even wood and its products need to be identified. So in that case, the technique that is used is DNA barcoding. So DNA barcoding, as I earlier mentioned, it is a DNA sequence marker. And DNA barcoding, it is used for, it was proposed for species identification in 2003 when Dr. Paul Herbert, he suggested that some regions within the DNA, they will not vary within the species. So in that species, that particular part of that DNA will always remain the same. It will not vary due to combination or mutation or such reasons. And those regions are mostly DNA segments which are outside the nuclear DNA. That is, which is the DNA which is present in the cytoplasmic organelles like the mitochondria and the plastids. So these organelles also have DNA, and the DNA within the mitochondria and the plastids also play an important role in the survival of the cell, in different functions of the cell. However, these organelles are always maternally inherited, and recombination does not happen in these organelles. Just DNA replication happens, and then they are divided between the daughter cells. So because recombination does not happen, he proposed that the regions within the mitochondria and the plastids are highly conserved, and a few of those regions were identified which are within the species which are highly conserved. However, those regions, when you compare with other species, there will be some kind of nucleotide difference. So this principle is used in DNA barcoding. And that region which we say is highly conserved is called the DNA barcode. And in animals, this barcode region is actually a 600 base pair segment of the mitochondrial cytochrome oxidase 1 gene. Whereas in plants, it is the RBCL, the ribulose bisphosphate, the large subunit code encoding gene, and the maturase K encoding genes within the plastids of plants. And apart from that, the intergenic spaces TR and H, PSBA, and another ITS2 region are also used as barcode regions in plants. So what we do when it comes to DNA barcoding in wood samples is, first from the wood sample, the DNA has to be extracted. That is again a long procedure. And then when the DNA is extracted, we amplify only the barcode region in the total DNA that we have extracted using specific primers. And these primers are again universal primers. So they bind to those regions. Now in plants, as we saw, the MATK and the RBCL regions, the flanking regions are all, again, they are also conserved. So those regions are used for primer designing, and hence the primers are universal. So using those universal primers, we amplify the region between those primers. And when it has amplified meaning producing large number of copies, those the resulting products, the PCR products, are called amplicons. So these amplicons are again short DNA fragments, which are double-stranded DNA fragments. So they need to be sequenced. So the difference that we are going to identify between samples is the difference in the sequence, that is the difference in the nucleotide sequence. These A, T, G, and C, how they are arranged within that 600 to 800 base pair fragment of the amplicon that we have. Now, after sequencing, once the sequence is known, the sequence of your sample is compared with sequences in a database. And again, DNA barcoding, I would like to point out that a reference database is required for sample identification. And if a reference database is available, up to the species level identification can be done. Now this table what I have shown here is actually a percentage identity matrix where 7 samples, the DNA sequences are compared. So being a matrix, the diagonally, you see that there is 100% similarity. But when you, the red highlighted regions, if you see, the sample number 3 and the sample number 7, they also show 100% similarity. So they are the same species. This is how identification is done. The sequences are compared, and this comparison is done by multiple sequence alignment of your sample fragment sequence with the reference database sequences. This is how it is visualized, the multiple sequence alignment for those 7 sequences. They are visualized like this only. You have A, T, G, and C arranged in random fashion. And whenever there is a difference in the nucleotides, for example, at position 1400, in the first sample it is A, whereas in the fourth sample it is T. In all other samples it is C, which means the first and the fourth sample, there is a variation at that point. Same way, there are the variations at all the points will be recorded when an alignment is made, and that is how the percentage identity matrix is generated, and you get similarity scores. So there are 2 different databases available online, And these databases are actually updated by researchers worldwide as and when samples are sequenced, not only plant samples, all organisms, as and when their barcodes are sequenced, those sequences are deposited in these databases. They are the nucleotide database of NCBI and the bold systems. These databases, we also use specific softwares for comparison. So in the nucleotide NCBI database, we use nucleotide blast for comparison with our query sequence. And in using the bold systems database, we use the bold ID engine. Again, these applications are also provided by them only. All we have to do is sequence that short barcode region of our sample, and that sequence is uploaded. As the query sequence, it is uploaded and it is compared with the complete reference database. And when this comparison is done, also you can restrict to, you want to compare it with the only plant samples or only bacterial samples or only animal samples, or you want to compare it with which species, samples of which species, all that can be assigned by you. And after comparison is made, using the nucleotide blast, you get a percentage similarity, whereas in using the bold ID engine, you get a barcoding gap. And those are actually values, and based on those values only you ascertain what species it is. Now, if it is going to be 100% similarity, then it is going to be the exact same species. Now, if the percentage is below 100, then you say it is more likely that it is this species. And one disadvantage of this is, for your sample, for your timber sample, for that species, sequences have to be available in the reference database. And if it is not available, then you should have already made a local database containing reference samples for those species. So this is how DNA barcoding is done. And here at IWST, we use that technique for species identification. Thank you very much.